Hi everyone. Today we will be talking about African evolutionary history inferred from whole genome sequence data of 44 indigenous African populations, written by Fan et al. My name is Chloe and I will be presenting with Anusha, Andrew, and Lauren. For the introduction, I'd like to start off with some information about the main author, Dr. Fan. He got his master's degree at Northwest Agriculture and Forestry University in 2008 and his PhD from the University of Constance for evolutionary biology in 2014. Since then, he's been working on research. He is very interested in topics which include demographic history and local adaptation of modern humans, molecular mechanisms of phenotypic variation, and the evolution of structural variation in modern humans. So humans inhabited the Earth around 2 million years ago, but in this paper, we are focusing on modern humans who originated in Africa around 300,000 years ago and dispersed around the world around 100,000 years ago, where they went to other places like Europe and Asia. They talked a lot about modern humans, and those are just Homo sapiens, which stand for wise men, and these are the individuals who originated 300,000 years ago. In the study, they used data from 94 people that were from 44 indigenous African populations. This is a fairly large study, especially considering others such as the Warren et al. Paper, where they only used one subject. We've talked about SNPs in class, but I thought I'd mention it again. So SNP stands for single nucleotide polymorphism, and that's basically just when a single nucleotide along the genome is different from what it should be. It's also important to mention that this is not the first study done to sequence genomes of African individuals. However, other studies, such as the African Genome Variation Project, have done it on a lesser scale, so this study tried to make theirs more diverse to obtain better results. They use a phylogeny to show the relationship of African populations, and I also wanted to mention that large-scale analysis has only been available since pretty recently, but it has allowed our knowledge of subjects to rapidly increase. For the main point, I kind of thought that is just to be able to use genome sequencing of indigenous African individuals to be able to gain more knowledge about certain aspects such as genetic variation, demographic history, and the origin of modern humans. Africa has the most diverse group of individuals. Using genome sequencing, the study helped infer the evolutionary history of Africa that led to this. A question I think that they were probably trying to figure out was what will genetic sequencing of different African populations reveal? And a hypothesis that is possible is genetic diversity will be high due to the length of time Africa has been inhabited. There will be more genetic diversity and linguistic diversity in African individuals that disperse to different locations. So there is a knowledge gap that this study helped to fill. Studies have been done to sequence genes of African individuals, but the studies weren't large enough to provide a high coverage genome sequence and sufficient genetic diversity. This changed that by selecting an abundant amount of individuals and having them come from various populations. The study contributed to science by presenting a sufficient amount of data on indigenous African individuals to create genome sequences and infer the evolutionary history of Africans from that. Researchers were able to include aspects such as ethnic, linguistic, cultural, and geographic diversity into their study and find out more about the evolution and diversity of indigenous African populations.
So for methods, they analyzed high coverage whole genome sequencing data from 92 individuals from 44 indigenous African populations and used a comparative dataset consisting of 62 Eurasian individuals from 32 populations represented in the Simon's Genome Diversity Project, which is the largest dataset of diverse high-quality human genome sequences. Next slide. Genetic variants for single nucleotide polymorphism and structural covariance were analyzed and compared across groups. They filtered the SNPs in linkage disequilibrium to minimize the impact of missing data. Linkage disequilibrium in the frequency of allele association being different from what's expected is a great way to make the study more relevant. SNP were used to construct phylogenetic relationship, and principal component and admixture analysis were used, which makes the study easier to analyze each genetic variations. Effective population size and divergence time analysis were also used, and all these methods are further explained through results. Next slide. Genetic structure analysis indicated that among Africans, genetic ancestry is largely partitioned by language, as you can see in figure 1, which shows different languages, where the language is classified into four phyla: Afrostatic, Nilo-Saharan, Niger-Congo, and Cohesian. Next slide. Figure 2 represents the phylogenetic relationship of African populations constructed based on genetic similarity of 44 African and 32 Eurasian populations. Using neighbor joining methods, the tree assumes no admixture events due to the nature of NG analysis that they use to build this tree. Also, another reason is that it's really difficult to do the study with considering admixture events. This means that they are assuming no interbreeding between individuals from two or more previously isolated populations, meaning recent common ancestry or experienced gene flow is being around clusters near the branches of this tree. Bootstrap values are indicated, which indicates the confidence for each node. Bootstrap values of the most nodes are greater than 90, which indicates that the resolution of construction is very excellent and it is strongly supported by the genetic data. Blue dots represent their certainty and red dots are represented for the bootstrap values which are greater than 90. Significance of building such a phylogenetic relationship is that we can compare the linguistic divergence to the genetic similarity. For example, the two Nilo-Saharan speaking populations, Mercy and Dinka, which group into a single cluster and consistent with the archaeological data indicating that the migration of Nilo-Saharan population to Eastern Africa originated from a source population in southern Sudan. It also indicates that the San genetic lineage is basal to all modern lineages as shown in figure 2. Next slide. Population structure in African populations. So the focus of this section was to identify genetic groups which historically have sufficiently isolated and self-contained genetic subpopulation. To identify these clusters of subpopulations, they did a principal component analysis. The data is taken from different dimensions dimensions and each base pair is one dimension. So principal component analysis is a way of reducing that a lot of dimensions and make the study clearer. They ran a PCA function to identify the clear patterns in the data. They found that there were 12 PCA patterns that was really stood out. The biggest pattern they found was the separation of African and non-African populations. The second one that stood out was San population from the rest, and the other was separation of CRHG individuals from the rest and Eastern and Western CRG individuals. Next slide.
So to continue, admixture analysis is a method of inferring someone's geographical origins based on an analysis of their genetic ancestry. So what they did here is they did an admixture analysis of 92 African and 62 West Eurasian individuals. Each of the bars on figure 4 represent one individual. K represents the number of clusters. The bottom bar shows the language classification of each individual. The colors of the bars represent the proportion of inferred ancestry, and the different colors on the bar, on one singular bar, means that there is admixture that occurred, and it has Bantu associated ancestry, which widely spread across populations in Eastern and Southern Africa, Sahel-Sudan Belt corridor of bidirectional migration coming from Nilo-Saharan and Middle East, and overall it reflects the historical gene flow with increasing number of clusters and shows divergence. Next slide. So to continue, multiple sequentially Markovian coalescent method was used, and it found that the NE, which is the effective population size, diverged around 200,000 years ago. And there was a common population bottleneck that occurred between 200,000 years ago and 60,000 years ago. And the group San maintained the largest NE in this period, which is figure 5A. And the CRHG populations maintained a relatively large NE as well. And this was consistent with higher level of genetic diversity compared to other sub-Saharan populations. Niger-Congo speaking and Nilo-Saharan speaking populations had lower NE, and the Afro-Asiatic speaking populations in North Africa had the lowest NE in figure 5D. Next slide. Continuing, the relative cross coalescent rate analysis, also known as RCCR, models the genetic separation between populations by the ratio of within and cross population coalescent rates. The big red line on the figure indicates a 50% coalescence, and it was estimated that ancestors of the San, Niger-Congo, Nilo-Saharan, and Afro-Asiatic populations were substantially diverged 120 to 100,000 years ago. And the earliest evidence of population divergence seen when RCCR is less than 1.0, and you can see that in figure 6A, which was approximately 200,000 years ago. Next slide. This table basically explains the divergence times estimated between African populations speaking languages belonging to the main language phyla, and all the estimates were inferred with MSMC using one individual from each population. The divergence times that were reported here were based on the relative cross coalescence rates at 50%. Next slide. And the last thing I'd like to talk about is the local adaptations. There were 6 meta-ancestry groups that consisted of 52 individuals. And in order to see these adaptations, the D statistic was calculated, which is basically a sum of normalized pairwise FST between a focal group and all other populations. It was computed for all SNPs with no more than 20% missing data in any group to identify these candidate regions of local adaptation. Variance in the top 0.1% of the empirical distribution were considered outliers. And so another tool that was used was the genomic regions enrichment of annotations tool, also known as GREAT. And it found that genes related to immune function were enriched near outlier windows in these populations. It also found that enrichments near genes that played a role in bone morphogenesis and near genes that play a role in renal and pancreatic development in the SOM populations. However, this could be due to low water availability in their diet. And lastly, the CRHG populations had enrichments near genes related to abnormal thyrotrophic morphology. And this is potentially proposed the same connection between thyroid and pituitary function and their short stature.
So overall, this study was able to identify over 26 million SNPs and use 7.5 million for the analysis of these populations. The authors determined that the ancestors of present-day lineage being of present-day populations developed substructure as early as 200,000 years ago, with the San genetic lineage being basal to all other modern human lineages that developed. Additionally, they were able to identify signatures of multiple waves of migration and pinpoint local adaptations and positive selection. Adaptations were found primarily at genes that involve muscle development, bone synthesis, reproduction, immune function, energy metabolism, and cell signaling. So to support these findings, the researchers used many different methods. They're able to construct a phylogenetic tree using the identified SNPs and ancestral allele. They did so assuming no admixture and produced highly significant results with bootstrap values over 90. They also used the MSMC method, which is a software used for inferring population structure through time from genome sequences in order to identify divergence. They found that the SAN maintained the largest NE value, which is effective population size, supporting their conclusion of the SAN being basal and also revealing other divergence patterns. Their PCA analysis went to identify patterns of migration with the addition of an admixture analysis, which revealed gene flow between Western Eurasian populations and North African populations. FST was then used for analysis of local adaptation, identifying adaptation in genes involving many biological processes. So overall, they used many statistical analysis to examine their data and used all of it to pull together a bunch of interesting information about the beginning of human evolution and its subsequent. As research study with more individuals to get specialized and complex coverage of the genome in order to analyze demographic history. Additionally, they mentioned taking a more specific approach to targeting regions of the modern human genome that seem to be introgressed from archaic, meaning Neanderthal pairings. This data could give us a better idea on more specific divergence times and population structures of early humans. Finally, the authors recommended looking into direct phenotypic genomic comparisons between contemporary and ancient samples in order to get a better idea of the formation of local adaptations and how complex traits evolved in the human population. So that is our presentation, and we will see you guys on Friday for discussion.